This is the Master Brewers Podcast, brought to you by the Master Brewers Association of the Americas, a volunteer organization dedicated to continually improving the products and processes of our membership since 1887.
Let's go!
Master Brewers brings you interviews with the industry's best and brightest in brewing science, technology, and operations.
Calm down, I'm moving too fast.
Support for this podcast and the following messages come from our friends at the Paul Mueller Company and Steiner. Please thank them. This podcast wouldn't be possible without their generous support.
Paul Mueller Company has been manufacturing quality brewing equipment since 1964. Our innovative design and engineering will save you time, labor, and ingredients, sending money back to your bottom line. Learn more about our new mobile hop module at paulmueller.com. This Master Brewers podcast is proudly sponsored by Steiner, a global leader in the hop industry focused on quality, sustainability, and innovation in new hop varieties and hop products. Contact our brewery sales team to provide you with the hop-related tools you need to craft your next great beer. For more information, visit hopsteiner.com.
I knew that it was not going to be too far of a stretch for us to develop yeast that were capable of making those flavor molecules during fermentation. The linalool synthase that was identified in mint was the best enzyme when we ported it over to yeast.
This week on the show, genetically modified brewer's yeast that makes hoppy beer without hops. Your recent publication in the journal Nature Communications made quite the splash in the brewing industry. The story was picked up by the New York Times, NPR, and countless news outlets with headlines like, "New beer yeast could make hops irrelevant," "GMO yeast mimics flavors of hops," and "Brewing craft beer with CRISPR, not hops." I've got to say, never have so many people forwarded me so many articles about a single publication. Charles, your inbox must be bursting at the seams.
We got more interest in the article than we expected, that's for sure. We're humbled by it, and we're also really grateful for the exposure. Some of the discourse was really useful and captured our sentiments, and others maybe didn't. But yeah, we're really excited that people took an interest in it.
Fair enough. Let's start off by explaining where hop flavor and aroma come from. Talk a little bit about essential oil and terpenes.
Yeah. Monoterpenes and sesquiterpenes are a class of molecules that are made primarily by plants, and they impart flavor and aroma, and in the case of hops and beer, they impart intense, like hoppy flavor and aroma to beer if they're added late in the brewing process.
And you already had some history working with terpenes, but not in brewing, right?
That's right, yeah. So we were interested in terpene biosynthesis from a biofuels perspective. So before I started this project, I was working on developing microbes that were capable of biosynthesizing other terpene molecules that would potentially serve as biofuel molecules or biofuel precursors. And it was only by chance that I came across this literature on what the primary flavor determinants of hops were. And so when I found out that linalool and geraniol were some of the primary determinants of cascade, I knew that it was not going to be too far of a stretch for us to develop yeast that were capable of making those flavor molecules during fermentation.
When did that first occur to you that it might be possible to engineer yeast to produce those terpenes that hopheads love?
Yeah, no, I remember. I was reading, one of my buddies that I homebrew with dropped off a book with me, or left a book with me by a guy named Fix called Principles of Brewing Science. And I was reading that book, and that's where I first saw linalool, geraniol, and I knew those molecules looked real familiar. So I sort of started thinking, well, maybe I should just test to see whether I can put these enzymes in a common lab yeast, which is it's actually somewhat far removed from Brewers yeast. They're just like the workhorse of sort of academia and like a lot of medical biosciences. So anyway, I put a few enzymes into the lab yeast and found a couple of enzymes that worked really well when they were expressed in lab yeast. And so that was actually a summer research project by an undergraduate at UC Berkeley that I was mentoring. And so while I was working on biofuels research, she came to the lab and sort of worked on that as a side project, and it worked better than we expected, and we got some really encouraging results. And that's really when we decided to push forward with actually trying to engineer industrial brewer's yeast.
Hop chemistry is complicated. There's still a lot we don't know, but we do know many of the compounds that are responsible for hop flavor and aroma. You focused your work on two specific monoterpenes for good reasons. Tell us more about linalool and geraniol and why you started with those.
So I'm reading this this book about brewing science, and it says linalool and geraniol are primary determinants. Well, there's a really long history of research that established those two as primary flavor determinants. So what I next did was I really dove into the literature, and there are dozens upon dozens of research articles that attempt to establish a relationship between certain molecules and flavor properties. There are some review articles that summarize this very well, but if you go back to the primary literature, every different study seems to point to a different collection of flavor molecules. But with these dozen upon dozens of studies, certain ones begin to emerge as repeatedly important. And linalool seems to be important for driving hop flavor in many different cultivars. And then there's some mixed reviews of how important geraniol is, but certainly it looks like for Cascade hops, geraniol is really important for driving its flavor. And then I think something you alluded to is that there are many additional terpenes. Some of them have, most of which come up in some studies, but not all studies. So I would say, while linalool and geraniol are perhaps the most firmly established as strong drivers of hop flavors, at least for Cascade hops, these other sort of more peripheral molecules are almost certainly important. Many of them are almost certainly important, but just there's not quite as much, quite as strong data supporting their involvement in dry hopping.
Makes sense. It sounds like the genes responsible for producing linalool and geraniol in hops haven't been identified yet. Did I get that right?
More or less. It's there has been some interesting research on basically where they've done some genome sequencing of hop. And there are a number of terpene synthases that have been found. But none of them have been established as committed linalool or geraniol synthases. And one thing I'm sure you're familiar with is that while linalool and geraniol are, they are drivers of hop flavor, there are other monoterpene molecules that are in much higher abundance in hops. So, for example, myrcene sometimes can account for, maybe Brian will check my numbers on this, but it can be like 50% of hop oil, at least 30%. Whereas linalool and geraniol may be less than 1% total. It's just that because they're more hydrophobic, they end up making it into beer at slightly higher abundances. And again, Brian, if you take issue with anything I'm saying, feel free to jump in.
No, you're pretty good there.
That's Bryan Donaldson speaking. He's the Brewing Innovation Manager at Laguintas Brewing Company. Bryan is one of the authors of the article we're discussing, and I should also mention that he's president of Master Brewers District Northern California.
I was just thinking about myrcene, and one thing to notice is you can't necessarily look at hops themselves. You have to look at what survives into the beer. So what actually makes it from the plant material into the finished product? While myrcene might be, and 50% is not a bad number to put out there, 50% of what you get out of your raw hops, what actually makes it into the beer is oftentimes going to be those terpenes that he's talking about, the geraniol and the linalool, because they do have, I believe, they're terpene alcohols. They're a little more soluble into the liquid.
Okay. So, Charles, without having sort of the genes responsible for producing those molecules in hops, without them being fully identified, that sounds like a pretty big hurdle. How did you approach dealing with that in your project?
Yeah, good question. So, what we did was we basically looked around in the literature, and of course, hops is not the only plant that makes linalool and geraniol. These are somewhat ubiquitous in the plant kingdom, and so we basically went and scraped through a bunch of literature and found papers where people had reported and other enzymes from other plants that make linalool and geraniol. And the one we ended up finding that the linalool synthase that was identified in mint was the best enzyme when we ported it over to yeast. And maybe in retrospect, this is not a total shock, whereas what we were talking about before with hops being 50% myrcene and less than 1% of linalool. On the other hand, if you take the extract or the essential oil rather of mint plant, it's like 90% linalool and linalool acetate, which is a derivative of linalool. So it's that those plant cells are really cranking out linalool. So part of what's going on, I suspect, is that the linalool synthase from mint plant just is a more resilient and robust and more active enzyme. Yeah, just to sort of go back to your original question, we ended up looking at candidate linalool synthases from 6 different plants, and the one from spearmint ended up working the best. We repeated something similar for geraniol synthase as well, and the one we ended up identifying as a solid candidate was the geraniol synthase from basil, actually.
Very cool. You knew from analyzing Hoppy commercial beers that the goal was roughly 0.2 milligrams per liter of linalool and geraniol, but how in the world were you going to hit that target?
Oh man, that's so glad you asked that question. Not very many people ask that question, and that was probably the biggest challenge that we had to face in this whole thing, because when you take enzymes from plants like mint and basil, and you stick them into yeast, you don't know exactly how much linalool and geraniol they're going to end up producing. So, it could be 0.02 mgs per liter. It could be, 2 mgs per liter. So we ended up taking those 2 terpene synthase genes and combining them with a couple other yeast genes that are involved in the terpene biosynthetic pathway. And we took those 4 genes and we added different combinations of promoters. So let me just back up for a second. Promoters are just probably many of the people in your audience know this, but promoters are just the genetic elements that control the timing and amount of the gene being expressed into a protein. So by choosing different combinations of promoters to drive those 4 genes, that results in different brewer's yeast strains with different concentrations of each of those enzymes and different timing under which those enzymes are expressed during the brewing process. So we started by testing 18 strains, each with a different combination of promoters driving those 4 genes, and then testing them in very, very small fermentations that we tried to set up such that they would mimic the industrial brewing process. So we, just to give you a sort of, entertaining to think about from a brewing perspective, because we were basically taking 30 milliliter cultures and equipping them with little airlocks and allowing those to ferment, 10% malt extract, and letting them ferment for 5 to 7 days, and then measuring the final concentration of linalool and geraniol in those resulting cultures. And so, like I said, we first tested 18 different strains and the way that we chose which promoters we would use to drive the expression of those 4 genes was somewhat random. We wanted to make sure we got a good spread from sort of low expression to high expression. And just a quick side note, all those promoters are genetic. So these genetic elements are from other yeast genes. That is, they are the genetic elements that drive expression of other yeast genes. So we're basically just taking them and repurposing them for expression of these terpene synthase genes, etc. Okay, so we tested those 18 strains, and very encouragingly, they ended up producing a range of different concentrations of both linalool and geraniol, and that range covered the 0.2 mg per liter target concentrations that we were hoping for. So that's sort of how we went about doing it. We tested more than 18 strains, but I think we might be getting into that in a second.
That's great. All right, so my wife keeps telling me that most people still don't know about CRISPR, and she's usually right, so we should probably talk about CRISPR. How are we going to do this? Should we just have everyone go listen to Radiolab or something? Or Brian, are you still there? Do you want to explain CRISPR? How about this?
I think it's best if Charles takes that one.
How about this? We don't need Radiolab. Let's take turns. I'll say a word, then you say a word, and I'll start, okay? Revolutionary.
Technology.
Groundbreaking.
Genetic.
Engineering.
I already ran out of words.
I wanted to end with scissors, but anyway. All right. Nah. So I think Charles, it's on you. You're going to have to give us the elevator pitch on CRISPR.
Yeah, no problem. So the thing that's really cool about CRISPR-Cas9 is that for brewing yeast, especially, is that so let me just take a step back and try and start at the beginning. So CRISPR-Cas9 is basically like a genetic defense mechanism that certain bacteria possess. And it was discovered by a whole host of different people, but some of the initial biochemistry was done in Jennifer Doudna's lab here at UC Berkeley, just around the corner from where we're at now. And basically, the implications of this discovery have been really interesting. Basically, it allows you or a scientist to make targeted double-strand breaks in DNA. And what that basically means is, let's say there's some part of the genome where you want to cut. You can do that practically anywhere in the genome. And then when you make that cut, the advantage of that is that you can oftentimes, typically you'll supply an exogenous piece of DNA, and if that DNA contains what's called homology, or DNA sequences that are similar to the cut ends, then that new DNA can be incorporated into the genome of the organism. And so does that, am I missing anything? Does that at least starting to make sense a little bit?
That sounds pretty good to me. It wasn't quite as good as Radiolab, but it was pretty good.
Okay, good. All right, so the reason this is so interesting for brewer's yeast is because, as everyone knows, brewer's yeast are tetraploid. And so the old school methods of introducing DNA into yeast genome just relied on basically trying to, the chance that a piece of DNA, without Cas9, the chance that a piece of DNA would incorporate into a given region of the genome is like 1 in a billion. I might be off by an order of magnitude there, but something on that order.
Right.
And so it's tricky to incorporate a piece of DNA into a genome. And what's really hard is to use that method to incorporate 4 pieces of DNA into 4 different loci, right? So if you're a tetraploid, maybe if you get 1 in, that ends up being genetically unstable, and it's not going to work for generating a new industrial brewing yeast that has that will stably contain the DNA of interest. When you introduce Cas9, you can make these targeted double-strand breaks, and that massively increases the rate at which a piece of DNA will incorporate into that particular genomic region. So it basically makes the numbers game possible that you can actually incorporate a piece of DNA at all 4 copies of the genome, all 4 loci of the genome. And that allows you to make genetically stable industrial brewers yeast strains.
Coming up.
And he let me know there was this project going on at Berkeley having to do with a genetically engineered yeast to make hop flavors, and was wondering if we were interested in running some sensory for it.
I'm John Bryce, and you're listening to the Master Brewers Podcast from the Master Brewers Association of the Americas. Here's what's coming up on the Master Brewers calendar. District St. Louis meets April 19th at Four Hands Brewery. The 57th District Caribbean Convention is April 19th to 22nd in St. Lucia. Don't miss the Draft Beer Basics and How Gases Affect Beer webinar, April 23rd. District St. Paul, Minneapolis meets May 10th. Need to build a food safety program for your brewery? Not sure what's required by law? Register for the Hazard Analysis and Critical Control Points course May 10th in Philly. District Philly meets at the New Yards Brewery the following day, May 11th. District Northwest meets in beautiful Hood River the weekend of May 18th. And District Rocky Mountain holds its Technical Summit in Fort Collins May 19th. View the full calendar of events at mbaa.com for more details or to find a district meeting near you. Now back to the show. Why don't you kind of lay out the remaining steps in your journey leading up to sort of the part where we can properly introduce Bryan and get into some of the tasting stuff?
Yeah, right on. So after we generated 48 strains that had these 4 different genes, all driven from 4 different yeast promoters. We basically took about a dozen of them that produced in the range of that 0.2 mg per liter sweet spot of both linalool and geraniol, and we started doing scaled up fermentations with those. And I think what we did at first is we just performed 1 liter fermentations with that group of a dozen different strains. We measured how much linalool and geraniol they were producing in the scaled up fermentations. And then eventually, we smelled the cultures and saw sort of how they. And then, obviously, that's not in the paper, but fun facts anyway. So we took 3 of our favorite strains over to UC Davis. We were able to collaborate with Dr. Charles Bamforth at UC Davis and his head brewer, Joe Williams, who's a PhD student in his lab. And Dr. Bamforth is actually how we ended up connecting with Bryan. But so what we did in Charlie's lab was we took those 3 strains as well as the control strain, and we made 40-liter batches of beer in his industrial pilot brewing facility. Joe, like I said, Joe Williams did that work. And so it was totally, professional grade beer. So we were obviously thrilled to be able to do that. And then Charlie suggested we take these samples over to Bryan at Laguintas, where he would be able to do the sensory, and obviously we're going to get into that. So that's pretty much how that ended up happening.
Very cool. So, Bryan, how about, hopefully you're still here, how about telling folks who you are and how you first heard about all this?
Absolutely. So I was contacted out of the blue one day by Charlie Bamforth, who I got my master's degree with at Davis back in 2011. And I had done my research there on the sensory of hop aromas, specifically generating new descriptors, and had somehow parlayed that into a job at Laguintas, running our sensory program. And then as of now, I'm doing R&D brewing, but still heavily involved in sensory. And he let me know there was this project going on at Berkeley having to do with genetically engineered yeast to make hop flavors, and was wondering if we were interested in running some sensory for it.
Were you skeptical at all? I feel like this is the kind of thing where I'd be pretty skeptical at first, but I don't know, maybe brewing innovation managers don't get skeptical. I don't know. We could never afford one of those at any of the breweries I worked at.
I am skeptical, but my background is in biochemistry, so I understood a bit of what this technology was and where it was coming from. And as a quick aside, I'll let Charles know this, but back when I was first going to grad school, my sister, I was talking to her, and she's a neuroscientist. She goes, "Bryan, we should start a brewery. You'll make the beer, and then I'll make the yeast, and we'll make them make all different flavors." And so I get this email from Charlie and Charles about that exact process and prospect, and I go, "Well, I have to do this work now because it's destiny." So we got on the phone. I think the first thing we did is I had a call with Charles and his team, and they just let me know what exactly it was they had done and what they were looking for. I ran it through the flagpole here at Laguintas and said, "Hey, I'd like to do some sensory for these folks from outside." Got all the approvals on both sides. Just the word of mouth at Laguintas, the IRB through Berkeley, and we did some sensory work for them.
So how did you approach that? Like how did you decide to set up the sensory work, and also why didn't you convince Charles to publish in the TQ? Huh?
That was our next target, was the TQ.
Yeah, I'm sure.
He was already on top of it. No, as far as running the sensory, really, there was a couple ways we could go about it, and that kind of is always the situation with sensory work. And for what they were looking for, we decided to just do difference testing. So we decided to see if these 3 strains of yeast they made the beer made with them tasted appreciably different from a beer made with the control strain, which was just the parent strain that they used and fermented the same wort out that Joe made, as well as the 3 strains. And so knowing we wanted to do a difference testing, there's a few ways you can go about that. Most people would just do a triangle test. But at this point, you're trying to triangle test between a control and 3 different beers. You got to do 3 different triangle tests. It's just a big mess. So we did a technique that I learned, and I'd heard about in school, but really had only started practicing about a year before, called difference from control. What a difference from control test is, you take all 4 beers, you have your control, you give the panelists that control beer, and you tell them, "Here's your control." And then you randomly label the 4 samples again. So your 3 variables and your 1 control. So you're giving them 4 blindly labeled samples, and you're saying, how different are each of those 4 from the control? The reason you leave a control in there blindly is so that you can gauge what sort of cognitive bias there is. When you tell people, how different is this beer, they're going to look for a difference even if there isn't one there. So that's what we did that way. And then you run the statistics, which honestly, I just let my computer do, and it'll tell you if it's significantly different from the control. And we found that 2 of the 3 samples they brought that first time were significantly different. We asked overall difference and difference in hoppiness. And they were different. There was definitely different flavor characteristics coming out of those beers that they brought.
Okay. Anything else you'd like to tell us about the sensory panel or the hop flavor and aroma from those beers?
Absolutely. So the panel was made up of just Laguintas employees. Anybody who wasn't driving that day, a forklift or a delivery truck is able to come to taste panel. So we got pretty good numbers, and we had a wide range of departments and styles represented. So, you had quality, you had brewing, you also had marketing, and you had the tap room. You had office folks, accountants at the taste panel. I can only really give my personal notes on the beers. I believe my quote that's been circling around is definite notes of Fruit Loops and orange blossoms, and I stick by that. Definitely got that. The one piece of feedback I really had for Charles is there's good fruity type aromas and flavors in there, but there was something fundamentally missing from the hop flavor. And that makes sense given that there's hundreds to thousands of aroma compounds present in these hops and in the finished beer, and trying to replicate the full scale with two molecules isn't going to work. But it seemed to me that there wasn't any of the vegetal character that you get from hops, right? Hops are mostly vegetal matter with some flavor compounds. So that's going to affect things in a way. But overall, it was really interesting, the flavors that we were getting out of it, and the fact that they were noticeable and they were distinct.
That's awesome. Charles, one of your claims is that yeast biosynthesized monoterpenes are more consistent versus conventional means. I'd like to hear both you and Bryan talk about that.
So my thinking on that is just the inside of a fermenter is a whole lot more controlled than a hop field that is, going to experience constant difference in just a bunch of different environmental factors. Could be the sun, could be the soil. So what we ended up looking at is we tried hopping a bunch of different beer with various preparations of Cascade hops, and we saw a pretty impressive range of concentrations of both linalool and geraniol. But what we found is that a skilled brewer like Joe could make the same beer with the same concentration of linalool and geraniol on different days with different, brewing processes. So we think that there's no doubt this will be more consistent than going the agricultural route.
And those preparations were from different farms, right?
That's right. Yeah, we took preps from I think a couple farms in Washington, a couple ones in Oregon, and one in Idaho.
And I'm sure Bryan could tell you all, he's probably told you all about hop selection and what all that's like, so I'm sure it makes total sense. So Bryan, what are your thoughts there on sort of that consistency issue?
Well, brewers don't have the luxury of using terroir and vintage with their beers, unlike some of the other local alcoholic beverage industries around here in Sonoma County, to remain nameless. And so this struggle can be from year to year getting consistent flavors out of changing crop years of hops and malt, but specifically we're talking about hops. And so I would have to agree from my scientist side that a fermenter, once it goes in there, you're controlling the temperature, you're controlling the pressure, you're controlling all of that. So theoretically, if you're controlling that well, your yeast should behave pretty similarly every time, and you should get more consistent results. I will say some of the feedback I've gotten is that doing that takes away from the brewer's art. I've heard from some brewers who say that this is taking away from what it is that brewers do and what makes brewers so special. And I have to disagree, because there's still so many subtle influences that the brewer has on the final product that can't be undone just from one ingredient. To quote Jeremy Marshall, my brewmaster, is it's not replacing anything, it's adding another color to the palette. So what we see happening is not necessarily that we're trying to take away from hops. I don't think hops are disappearing anytime soon. At least that's what we told our hop farmers when this paper came out. But is there something we can add with what Charles is doing that makes something really interesting? And that's what excites me as a scientist and as a brewer.
Well said. Brian, even though it's not well understood, some brewers feel that biotransformation of hop compounds during fermentation is important to their process. Those brewers report preferred sensory results when dry hopping prior to the end of fermentation, but then historically that's created some other logistical headaches like trying to harvest dry hopped yeast slurry. Brian, from your perspective, how attractive is the idea of getting these terpenes into the fermenting beer without having to separate out spent hop material from beer or yeast?
It's definitely intriguing. I would definitely say at Laguintas we ascribe to biotransformation being very important for the flavor of our beer. We haven't necessarily done any specific controlled experiments, but through years of tasting, when we don't hit our timing for dry hop, we notice a difference. I think there's just in the historical and traditional sense, there's ways around that. You don't necessarily have to harvest dry hop yeast. You could do a harvest right before you dry hop or whatnot. But if there's a way that you can liberate some more of these flavor compounds using less yeast or just having the yeast make some of them, that's really exciting. Charles and I both got to attend the hop conference last year in Oregon last summer. And I think we both came away with a lot of ideas of how technology like that he's working on could be used beyond just generating a single flavor compound, but enhancing that biotransformation possibilities or bringing in new flavor compounds that might not be liberated from the hops very efficiently at this point, given what we have. So to answer your first question, it's exciting to maybe not have to deal with mixed hop yeast slurry, but just everything else that it could provide is interesting.
Great.
Yeah, and I just to sort of reiterate Brian's points, I just want to say, like, the way we think about this, having discussed this with brewers and folks in the industry, is like, this is similar to what Jeremy was saying, another color on the palette. It's like, we think this is just another tool in the toolkit. And ultimately, the way we're thinking about this is just, how can we do more with less? So that's sort of that's our approach.
Your article made the important point that full flavor imparted by traditional hopping is likely to rely on a more diverse bouquet of molecules. I can only imagine you've been working on engineering yeast to produce various packages of hop aroma compounds since you submitted this article last August.
Yeah, that we sure are.
And I suppose it makes the most economic sense for you to target aromatics from some of the highly demanded proprietary cultivars such as Mosaic and Citra and Galaxy and so on.
Yeah. I mean, I think, I think this sort of come up both from my end and from Brian's end, is that, like, we're interested in developing this technology as a tool for making better beer. And so, this initial project serves as a proof of concept, showing that this technology can be, reduced to practice. But I think that what's more important is it just shows I think it's just exciting because it shows that we're really just scratching the surface of what yeast is capable of doing. So, looking towards the future, we're really excited about new flavors that are reminiscent of hops, that'll just make better beer than what's previously been available. It's just getting back to the point of just, we envision this just being another tool in the toolkit to make better beer.
To jump in real quick, I think there's almost an infinite number of different combinations that Charles's team could work on. If you look at it from a brewing side, from a hop growing side, there's not only differences between cultivars, of which there's probably a few hundred these days, but there's differences based on where they're grown, as he showed. There's differences based on when they're harvested. Now, breweries will pick their specific hops based on not only cultivar and location, but when they're harvested. Some like to harvest them earlier when they have a certain characteristic versus some that like to harvest them later. That same varietal can go through, 3 or 4 different distinct phases through its ripening cycle and get different characteristics. So there's a lot of possibilities to really pinpoint exactly what you want.
I'm going to read a quote from your article. While historic consumer trepidation towards genetically engineered foods is of concern for widespread adoption, the general increase in consumer acceptance of such foods when tied to increased sustainability is encouraging, end quote. Charles, do you think the consumer really cares if the yeast that made their beer taste hoppy was produced with magical genetic scissors?
I think in some cases people care. And I think it's interesting. I think in some cases people are going to look negatively on using magical genetic scissors to make their brewer's yeast. But I think in some cases people are going to look positively on it. And one of the things that we try and emphasize in this is that by doing more with less as far as hops go, we can make the brewing process more sustainable. And I think I mentioned at the beginning of the show, I come from a biofuels lab. Like, we're very interested, we're very committed to sustainability. We really believe in the green revolution. We really think that technology could be used to make industrial processes more sustainable. So I hope that strikes consumers as something that is an advantage that they would want to buy into, and I'm not quite as worried about brewers, to be honest, because I think brewers are way more science oriented types. Like, they are much more knowledgeable about these types of issues. So I bet if I go out and find 10 people on the street, maybe a few of them would feel some type of hesitation towards wanting to consume something that's used genetic engineering. But I think that if you go find 10 brewers, you'll be hard pressed to find one that's anti for the sake of fear of new technology or the like. I think brewers are going to be concerned about consumer response, but maybe they're the best people to be educating the consumers about the benefits of this technology. Yeah,
we want to use all the tools in the toolbox. So I think I think you're right there. Charles, are we going to see you guys at the Brewing Summit coming up in August in San Diego?
Heck yeah.
Okay. Are you presenting this work there? Or?
No. It's funny. I asked them if I could present it, but they said because it's been published elsewhere that they didn't want me to. So. Is that really?
You can present to our joint meeting with Southern California in February next year.
Yeah, you should do that.
That sounds great. We're also hoping to have some new science to talk about at the next summit, so we won't have to rely on this old stuff.
That was Charles Denby and Bryan Donaldson here on the Master Brewers podcast. If you haven't already listened, do yourself a favor and go download the Radiolab episodes about CRISPR. After that, go to mbaa.com and register for the Brewing Summit 2018 in San Diego, where you can drink beer and learn things with smart people like Charles and Bryan. 130 years ago, Master Brewers was built on the concept of brewers helping each other out so we could all make the best possible beer. That's still true to this day, and it's where a lot of the camaraderie in this industry originated. Master Brewers' award-winning Ask the Brewmasters is the best place to go for troubleshooting, where you'll find the industry's only discussion forum that's moderated for technical accuracy by a team of experts. See what everyone else is talking about at community.mbaa.com. Did you enjoy today's episode? Would you like us to keep making more? If so, there's a really simple way you can let us know. Subscribe, rate, and review the Master Brewers Podcast on iTunes, Stitcher, or wherever you get your podcasts.
Heading out to any other place. My fist full of courage, my heart full of rage. Well, I can't get stuck, I can't be losing too much, and then